Section 11.2, Mechanisms of Evolution. By the end of this section, you will be able to describe the four basic causes of evolution: natural selection, mutation, genetic drift, and gene flow, and explain how each evolutionary force can influence the allele frequencies of a population. The Hardy-Weinberg equilibrium principle says that allele frequencies in a population will remain constant in the absence of the four factors that could change them. Those factors are natural selection, mutation, genetic drift, and migration, or gene flow. In fact, we now know they are probably always affecting populations. Natural selection. Natural selection has already been discussed. Alleles are expressed in a phenotype. Depending on the environmental conditions, the phenotype confers an advantage or disadvantage to the individual with the phenotype relative to the other phenotypes in the population. If it is an advantage, then that individual will likely have more offspring than individuals with other phenotypes, and this will mean that the allele behind the phenotype will have greater representation in the next generation. If conditions remain the same, those offspring, which are carrying the same allele, will also benefit. Over time, the allele will increase in frequency in the population. Mutation. Mutation is a source of new alleles in a population. Mutation is a change in the DNA sequence of the gene. A mutation can change one allele into another, but the net effect is a change in frequency. The change in frequency resulting from mutation is small, so its effect on evolution is small unless it interacts with one of the other factors, such as selection. A mutation may produce an allele that is selected against, selected for, or selectively neutral. Harmful mutations are removed from the population by selection and will generally only be found in very low frequencies equal to the mutation rate. Beneficial mutations will spread through the population through selection, although that initial spread is slow. Whether or not a mutation is beneficial or harmful is determined by whether it helps an organism survive to sexual maturity and reproduce. It should be noted that mutation is the ultimate source of genetic variation in all populations. New alleles, and therefore new genetic variations, arise through mutation. Genetic drift. Another way a population's allele frequencies can change is genetic drift, which is simply the effect of chance. Genetic drift is most important in small populations. Drift would be completely absent in a population with infinite individuals, but of course no population is this large. Genetic drift occurs because the alleles in an offspring generation are a random sample of the alleles in the parent generation. Alleles may or may not make it into the next generation due to chance events, including mortality of an individual, events affecting finding a mate, and even the events affecting which gametes end up in fertilizations. If one individual in a population of 10 individuals happens to die before it leaves any offspring to the next generation, all of its genes, a tenth of the population's gene pool, will be suddenly lost. In a population of 100, that one individual represents only 1% of the overall gene pool. Therefore, it has much less impact on the population's genetic structure and is unlikely to remove all copies of even a relatively rare allele. Imagine a population of 10 individuals, half with allele A and half with allele little a. So one dominant and one recessive. The individuals are haploid. In a stable population, the next generation will also have 10 individuals. Choose that generation randomly by flipping a coin 10 times and let big A and let heads be big A and tails be little a. It is unlikely that the next generation will have exactly half of each allele. There might be 6 of one and 4 of the other, or some different set of frequencies. Thus, the allele frequencies have changed and evolution has occurred. A coin will no longer work to choose the next generation because the odds are no longer 1/2 for each allele. The frequency in each generation will drift up and down on what is known as random walk, until at one point either all capital A or all lowercase a are chosen, and that allele is fixed from that point on. This could take a very long time for a large population. This simplification is not very biological, but it can be shown that real populations behave this way. The effect of drift on frequencies is greater the smaller a population is. Its effect is also greater on an allele with a frequency far from 1/2. Drift will influence every allele, even those that are being naturally selected. Figure 11.7. Genetic drift in a population can lead to the elimination of an allele from a population by chance. In each generation, a random set of individuals reproduces to produce the next generation. The frequency of alleles in the next generation is equal to the frequency of alleles among the individuals reproducing. Genetic drift can also be magnified by natural or human-caused events, such as a disaster that randomly kills a large portion of the population, which is known as the bottleneck effect, that results in a large portion of the gene pool suddenly being wiped out. In one fell swoop, the genetic structure of the survivors becomes the genetic structure of the entire population, which may be very different from the pre-disaster population. The disaster must be one that kills for reasons unrelated to the organism's traits, such as a hurricane or lava flow. A mass killing caused by unusually cold temperatures at night is likely to affect individuals differently depending on the alleles they possess that confer cold hardiness. Figure 11.8, a chance event or catastrophe can reduce the genetic variability within a population. Another scenario in which populations might experience a strong influence of genetic drift is if some portion of the population leaves to start a new population in a new location, or if a population gets divided by a physical barrier of some kind. In this situation, those individuals are unlikely to be representative of the entire population, which results in the founder effect. The founder effect occurs when the genetic structure matches that of the new population's founding fathers and mothers. The founder effect is believed to have been a key factor in the genetic history of the Afrikaner population of Dutch settlers in South Africa, as evidenced by mutations that are common in Afrikaners but rare in most other populations. This is likely due to a higher than normal proportion of the founding colonists, which were a small sample of the original population, carried these mutations. As a result, the population expresses unusually high incidences of Huntington's disease and Fanconi anemia, a genetic disorder known to cause bone marrow and congenital abnormalities, and even cancer. Gene flow. Another important evolutionary force is gene flow, or the flow of alleles in and out of a population resulting from the migration of individuals or gametes. While some populations are fairly stable, others experience more flux. And many plants, for example, send their seeds far and wide by wind or in the guts of animals. These seeds may introduce alleles common in the source population to a new population in which they are rare. Figure 11.9, gene flow can occur when an individual travels from one geographic location to another and joins a different population of the species. In the example shown here, the brown allele is introduced into the green population.